So I want to talk a little bit about tRNA and its role in the process of protein synthesis. But before we get into that, let's just do a quick warm-up. So I'm hoping if somebody were to ask you the question, well, what are the two stages of protein synthesis? I'm hoping that you would say these two things in this order. You would say first is transcription. And then, secondly, we have translation. Well, in the last couple of lessons, we've talked about transcription. So let's just kind of take stock of where we are before we move forward and talk about translation. So you'll recall that we talked about how, if this is, let's say, a cell nucleus, and we know that our DNA is all located in said nucleus. And we have, maybe our endoplasmic reticulum. I'm terrible at drawing, but you get the gist of it. Endoplasmic reticulum all around the nucleus. And then located on the endoplasmic reticulum, somewhere in the cytoplasm, are our ribosomes. And so the problem is that our DNA can't leave the nucleus. So we need to have a way of getting DNA's code out to the ribosome. And so what happens in transcription is, a copy of the DNA of a particular gene is made in the form of mRNA. And then it can go to the ribosome. And that's where we are right now. Okay, we've talked about how mRNA is produced in transcription. Now we're ready to talk about how it's utilized in translation. So before we begin with that, I wanted to talk to you about specifically tRNA. So I've pulled up a couple of documents, so stay tuned. So I am going to post another video that will describe the process of translation, but I just want to orient you with respect to the diagram that I have here. So here is our mRNA strand. And so we can see very clearly the 5' end and then the 3' end. We look along here and we can see that we have uracil instead of thymine because it is RNA. And then we have those nucleotides grouped in pairs of 3 representing the codons. And so here we have our ribosome. And we can see, if you look carefully, that there are 2 subunits of the ribosome. You'll remember I talked about the larger subunit and then the smaller subunit. That will come up in a separate video. And what is bringing in our new amino acids is our tRNA molecule. And so that's what you're seeing sort of here. It almost looks like an upside down L of sorts. So here's one tRNA and here's another tRNA, hence the 1 and the 2. And so now let's talk about the tRNA molecule a little bit more. So you'll see some different ways that the tRNA molecule might be illustrated. So first of all, you might see sort of this space-filling molecule where it has that sort of L shape, and that's what you'll see in this diagram. You'll see it sort of almost looking like a double helix sort of twisty thing. Right? And often you'll see what is referred to as the cloverleaf shape of the tRNA, where you can see some different arms of it. Okay? So notice here that there's a 5' end and a 3' end. So I want you, instead of thinking about this as a double helix, think about it as a single strand that's kind of looped onto itself. Right? So 5', 3'. But along the way, how in its conformation, there might be some hydrogen bonding that's occurring there, and that's what we're seeing. That looks like a double helix structure when it's really not. So things to notice: we're not going to talk about the D loop and the T loop in this particular lesson at this stage of things. You will probably see that next year. But what you should take note of that is present in each of these diagrams is this thing called an Anticodon. So I'm going to bring up a different diagram to discuss tRNA and the Anticodon with a little bit more detail. Okay, so here I've pulled up an alternate diagram. You'll notice that in this diagram it's also represented in that cloverleaf form that we talked about. So two important parts of the tRNA that I want you to pay attention to, and number one is what we've already discussed, which is this anticodon business. And so I want you to imagine that, here is our mRNA strand, and what's not written, but we can sort of assume is there, is our ribosomes, the smaller and then, sorry, the larger and then the smaller subunit. And it is clamping our mRNA strand between those two subunits. Now, remember that, well, in this process, the mRNA codons are being translated into amino acids to make up our protein. Well, how does it do that? Well, the role of tRNA is to bring in those amino acids. But how does it know which one to bring in? Well, it turns out that the anticodon that is on our tRNA molecule is complementary to the codon that we find on our mRNA. So, for example, if our codon on the mRNA strand is GCC, then it should make sense that our complementary anticodon on the tRNA molecule would be CGG. Now, you should know that the amino acid that comes with the tRNA represents the codon and not the anticodon. So, for example, the amino acid that would be located on this tRNA, you would look on your codon chart to see what that would be, and it would be the one for GCC. So there it is. That is that's our tRNA molecule and how essentially it is able to bring in the correct amino acid. It does so by having that complementary anticodon. So look for another video that will outline the process of translation for you. And if you have any questions, be sure to reach out.